This video will be an introduction to chromosomes, and it is found in chapter 2 and 11 of your textbook. So genetics is somewhat unusual in that it is studied at multiple different levels. So we will talk about traits in the organism. For example, eye color or skin color. These are characteristics that are determined by the genes that we have in our cells, but we look at the entire organism to see that trait. We'll also talk about genetics on the chromosome or the cellular level. We'll look at the chromosomal organization of the genetic material in cells, and that chromosomal organization will be important when we talk about mitosis, meiosis, and when we talk also about how inheritance patterns are affected by the organization of genes on chromosomes. Then we'll also talk about genetics at the DNA level. We'll talk about DNA structure. We'll talk about how genes are arrayed upon that DNA. So when we look at chromosomes, things are very different between prokaryotes and eukaryotes. We will touch on prokaryotes from time to time in this course, but most of our time will be spent talking about eukaryotes. So prokaryotes include bacteria and archaea. These organisms are mostly single-cell organisms. They do not have a separate compartment for the DNA. They do not have a nucleus. And their DNA is found as a circular chromosome. Most of the time it's just one chromosome, but occasionally you can have multiple circular chromosomes. So eukaryotes include all plants, animals, and even some single-cell organisms such as yeast. So eukaryotes do have a separate compartment for the DNA, and that is called the nucleus. The chromosomes are found within the nucleus, and the chromosomes are multiple pieces of linear double-stranded DNA. One thing I'll just highlight now is that eukaryotic cells do in fact have a small amount of DNA that is found in their cytoplasm. There are small chromosomes that are found within the mitochondria of eukaryotes and also chloroplasts in plants. So if we think about how these different types of species divide and how they reproduce, prokaryotes divide by splitting, and both daughter cells are the same as the mother cell, and that splitting is called binary fission. Eukaryotic cells, on the other hand, divide in two different ways. First of all, by mitosis. This would be cellular reproduction for most cells, and this would be a case where one mother cell gives two daughter cells that are identical to the mother cell. Eukaryotic cells can also divide by meiosis, and these would only include particular cells in the species, only the germ cells that are getting ready to make gametes for sexual reproduction. So each eukaryotic species has a unique number of chromosomes. A few of these are listed here. So for example, fruit flies have a total of 8 chromosomes in almost every cell of their body. Humans have 46 total chromosomes, and the number of total linear chromosomes varies quite widely, from very few to very, very many. So butterflies and I think ferns are another species with very, very high numbers of chromosomes. One thing I'll highlight is that the number of separate chromosomes in a species really doesn't have any relationship to how complex that organism is. Something you might notice here is that all of the chromosome numbers are even numbers, and there's a reason for that. The reason that eukaryotic cells have an even number of chromosomes is because there are 2 of every single chromosome. So in order to be able to undergo sexual reproduction via the process of meiosis, you have to have 2 of each chromosome. The total number of chromosomes, so the numbers that are shown in this table here, is the diploid number of a species, or the 2n number. And it is referred to as such simply to highlight the fact that that total number includes 2 of every chromosome. The haploid number, which is called 1n or just n, is half the diploid number. And the meaning of that number is that it's actually the number of unique chromosomes, since there are 2 of each chromosome. So here's a figure that shows condensed chromosomes for a particular species of deer. It's called the Indian muntjac. This particular species has 6 chromosomes, and so therefore you count up the number of chromosomes, and that tells you what the 2n or the diploid number is. So 2n equals 6, and the haploid number equals 3. This cell is actually stained by a method called chromosome painting to show the different types of chromosomes in different colors. And you'll see that there are 2 green chromosomes, 2 red, and 2 blue chromosomes. So there are 3 different types of chromosomes. There is a relationship between the 2 chromosomes that are the same. So for example, if you look at these 2 green chromosomes, those chromosomes are called homologous chromosomes. For this particular individual, they inherited 1 of those green chromosomes from their mother, 1 of them from their father. Homologous chromosomes are similar to each other, but they are not identical to each other. They are similar in a number of ways. So they are the same size, roughly the same number of bases of DNA. They have the same genes on them. And those genes are found in the same order between these 2 chromosomes. So for example, if one gene, just say gene A, was found right here near the end of this green chromosome, you would also find a gene A near the end of the homologous chromosome. So they have the same genes, the same order of genes, but they might have different versions or different alleles of those genes present on those separate homologous chromosomes. So that's illustrated here in this diagram. So there are 6 chromosomes total here. That would be a 2n number equals 6, but several of these are homologous to each other. And so, for example, this chromosome here with the A, B, C, and D genes is homologous to this other chromosome with the A, B, C, and D genes. If you'll notice, there are the same genes, but they have different versions of those genes. So this chromosome has a dominant big A version of that gene. This one has a little a recessive version of that gene. So if we look at diploid organisms such as ourselves, most of our lifetime is spent in the diploid state. So when each of us were created via fertilization with an egg and a sperm, a single cell was formed, and that would have been called the zygote, or the fertilized egg. That one cell will give rise to the adult organism, and all of that growth that took place was via mitosis. If we look at the cells of our body, the cells of our skin cells, our muscle cells, our bone and our brain cells, all of those cells, if we could peer into them, we would see a diploid number of chromosomes in each of those nuclei. There's one exception to that, and that is the germ cells. So the cells of the gonad, the testis and the ovary, have the potential to undergo meiosis. And meiosis is this process whereby a diploid cell will undergo 2 divisions to create haploid cells. If you'll note, a haploid cell has 1 of each type of chromosome, and it has a 1n number of chromosomes. Once we form the egg and the sperm, we have the potential for fertilization to occur, and once fertilization happens, then we go back into this diploid state. I just wanted to highlight one side point. You may have heard of organisms that are haploid. Haploid organisms are in fact eukaryotic organisms, and they also have the same kind of change between the diploid and the haploid states. The difference between them is where they spend most of their lifetime. So here's an example shown over here. This is actually bread mold. It's a haploid organism, and they also go between this haploid and diploid state. So the main organism that you see growing in your bread and making it moldy is actually these hyphae that originate from spores. So those spores are actually gametes. They are the product of meiosis, and they are haploid cells, and they start to divide by mitosis. They can divide for quite some period of time, but at some point, if they are going to undergo sexual reproduction, they're going to have to become diploid. Once they decide to become diploid, they actually form these two different mating types, and those cells then of two different types will fuse together. And that nuclear fusion is creating now a diploid cell. In the bread mold, as soon as that diploid cell is formed, it's called a zygospore, it immediately undergoes meiosis and goes back to the haploid state. And so haploid versus diploid organisms really just indicates where that organism spends most of its lifetime. The last thing I'd like to go over is very briefly to review how eukaryotic DNA is packaged in the nucleus. So there is a lot of DNA in each one of our cells, roughly 2 meters of DNA, and we need some organization. The DNA is actually wrapped around these structures called nucleosomes. And a nucleosome is made up of 8 different proteins. These proteins are called histones, and there are actually 2 of each type. So H2A, H2B, H3, and H4. And that DNA actually wraps around a nucleosome 2 times. So you can see it goes around 1 time, it goes around a second time. The length of DNA that wraps around one nucleosome is around 146 bases. And this creates a structure that is 11 nanometers wide. At the next level of organization, each one of those nucleosomes is arranged in what's called a solenoid. So now a nucleosome are these little purple spools, and the DNA is wrapped around each one of those 2 times. And the solenoid is actually stabilized by this additional histone protein called H1. And the solenoid actually makes these kind of like a disc or a wagon wheel that then spirals around. A solenoid is actually the word for a spring, and so it's kind of like a slinky in that it is spiraling around. The diameter of this structure is 30 nanometers wide. And then there's 2 additional levels. Level 3 is actually a scaffold in which the DNA is looped onto a scaffold of proteins. The proteins are shown in red here. You can see these looped structures, and the structure that's shown is actually the solenoid structure. This produces something that is about 300 nanometers wide. And then the final level of organization is the supercoiled scaffold, which is actually 700 nanometers wide. And this is what we see when we have condensing of chromosomes that is observed during mitosis and meiosis. And so just to summarize, these are the different levels of DNA organization in eukaryotes. We have the naked DNA. It gets wrapped on these spools of histones, and then those histones kind of join together to create the solenoid or the slinky. That solenoid then arranges into loops, and those loops then get supercoiled when we have a mitotic chromosome. The density of DNA varies tremendously, and I wanted to introduce this early on in the course because we're going to come back to histones and nucleosomes and how important they really are. They're very important in regulating how tightly wrapped the DNA is, and that really strongly affects how that DNA is able to be used for transcription.